Okay, I guess we'll get started. Welcome to our friends. Sorry about the delay. There we go. Okay, this webinar is eligible for a 1 hour of SIIM IP credits that can be used towards your ABII, CIP certifications or recertification. Okay, so at the end, please, or during this talk, please add your questions using the Q&A icon in the Zoom menu throughout the webinar, and we'll work to answer them after the talk. Okay, so I'm going to introduce our speakers here. So I'm Angel Ngor, like I said, with Barco. The first presenter is Tom Kimpy. He's a VP of Technology and Innovation at Barco Healthcare. Together with his teams, he has contributed to many innovative products such as mammography, tomosynthesis, pathology display systems. He is an inventor of several patents and has co-authored tens of scientific publications in the domain of medical imaging and displays, visualization, optics, image processing, artificial intelligence. He regularly is a scientific reviewer, member of a program of committees of scientific conferences, and contributes to international standardization activities. Our second speaker is Brian Van Meter. He is a senior product manager at Barco. He is responsible for Barco's digital pathology solution portfolio globally and has been involved with developing workflow tools for digital pathology and diagnostic imaging. He serves as a member of the Digital Pathology Association Educational Committee. Prior to joining Barco, he was the global marketing manager for advanced visualization software at GE Healthcare. Agenda will cover the characteristics of digital pathology images, perception of digital pathology images, medical and non-medical display systems fit for use in digital pathology, and then close up with some conclusions and Q&A. So Tom, I'll kick it off to you.
Thank you very much, Andrew, for the kind introduction, and welcome everybody to this seminar, and thank you for joining. So let me start with explaining on characteristics of digital pathology images. If the slide wants to move, at least. Yes. So short introduction, what is digital pathology? Well, digital pathology covers the acquisition, management, sharing, and interpretation of pathology information in a digital environment. So many of you probably know anatomical pathology labs. What is happening in anatomical pathology labs is that specimens are put on a glass plate, they are stained with chemicals, and then these glass plates are viewed by pathologists to come to a diagnosis. So in the past, this diagnosis happened by looking to those glass slides under a microscope. What is happening today is that in digital pathology is that these slides are scanned by a scanner, digital images are created, and then these digital images are being reviewed on a workstation with a display system. So what is special about digital pathology images? Well, to start with, the resolution, the image resolution. So the exact image resolution, of course, depends on the magnification factor that is set by the microscope. Typical factors are, for example, a 4x or a 20x magnification. So if we take 20x as an example, such an image really has a huge resolution. It will be typically something like 80,000 by 60,000 pixels, or if you put that in gigapixels, 4.8 gigapixels. Now, this number may not say a lot, but maybe some of you know full-field digital mammography images. Well, for a comparison, a typical full-field digital mammography image is only 12 megapixel, not gigapixel, but megapixel, which means that actually one single whole slide image or digital pathology image is about 400 times larger than a full-field digital mammography image. Just to put it in perspective, here you see on the bottom the relative magnitude of a FFDM image, and then on the top right, a digital pathology image. So these are really huge images. Huge images, but with that amount of information, it's very clear that it's not possible to see all details of that image at a glance. So the human eye has a limitation in the visual acuity. So typically it's expressed in line pairs per arc minute. And then it's 1 line per arc minute, or you can also put it in degrees, 1 cycle per 0.017 degrees. Practically, it means that if you're about 25 centimeters from the display or from a target, you can actually see things that are 0.07 millimeters apart. So it's clear that digital pathology images go well beyond what a human eye can perceive. That has as a consequence that the only way to inspect the full slide is by doing a lot of panning and zooming operations, meaning that you zoom into the image, you inspect a certain sub-area of interest, you zoom out again, you pan, and so on. File format-wise, therefore, digital images, digital pathology images typically are using a kind of pyramid structure where different zoom levels are stored to be able to quickly visualize the information at different zoom levels. Another thing special about digital pathology images is that they are color images. Compared to radiology, which is still today mostly grayscale images, digital pathology is always color images. And actually high definition color images, because typically per color channel, there is a 10 to 16-bit depth in each channel. These images, digital pathology images, also have a very wide color gamut. To give you an example, if you take typical H&E images, and this is shown in the table and the picture on the left side, on an sRGB display, which is kind of the standard, it's impossible to show all the colors that are present in those H&E images. Quite a number of those pixels of that information will not be or cannot be rendered on an sRGB gamut. So you typically need a very large gamut in order to be able to show all the colors present in digital pathology images. This holds for H&E images, but it's even more important for IHC, so immunohistology stains, and for fluorescence stains. So you can ask then, why is that so important? Why is it so important that the color of pathology images is rendered and presented in a correct way? Well, I can answer that question by referring to something which very commonly happens in a digital pathology flow or in a general analog pathology flow. Quite a lot of cases are being referred to another lab or another pathologist for a second opinion. What we see in practice is that very often when the lab that the case is referred to is receiving the slide, the first thing that they are doing is asking for the block of the tissue, and they are cutting again and creating a new stain based on the procedures of that lab. And the reason is because if that's not done, there are quite apparent differences in color behavior between slides generated in different labs. And that creates a lower confidence in the quality of the slide and lower confidence in diagnosis. And that's why very often actually new slides are created. So this is true for human reading, for manual reading, but with artificial intelligence being used more and more, accurate colors is even more important because it's well known, and there are a few references that you will find here and in the reference section of the presentation. It's well known that AI algorithms are quite sensitive to changes in color characteristics. So in other words, if you give a certain slide image to an artificial intelligence algorithm, or you give the same tissue that is generated in another lab with a different color characteristic, it's likely that there will be slight or less or larger differences in the AI output. So color accuracy is quite important. Now I would like to say something on perception of images, digital pathology images. And this is a quite theoretical part of the presentation of the webinar, but it's a very important part of the webinar. And the reason is medical images are no holiday photographs, or they are no images that you look at and that you expect them to look in a pleasing way. Medical images are really images that are used to take life-mattering decisions upon, whether it's diagnosis, treatment, or so on. So it's very important that all the information, all the small details that are present in such a medical image can actually be seen by the observer, can be seen by the pathologist or the radiologist or the dermatologist or so on. So this is a subject field, a research field, which has been extensively studied in the past. And I just want to point you to the PhD of Peter Barton, who has done a lot of research on human visual perception, and that has been applied into radiology applications. So the question that was posed at that moment in time was, what makes an image good? How can you be sure that all the image details present in a radiology image are perceivable, can be seen by a radiologist? So the way that Peter Barton approached that was by doing a lot of psychovisual experiments. I won't go into the details, but a pattern that he used a lot were the sine wave patterns that you can see on the right bottom side. And he presented those sine wave patterns or Gabor patterns to a lot of observers, those patterns having different luminance levels, contrast levels, spatial frequencies, and so on. So it was quite an extensive research, and he came up with very interesting results that actually formed the basis for the DICOM GSDF, Greyscale Standard Display Function, which is standardized in radiology. So this is a little bit of the summary of what he came up with. He came up with the concept of just noticeable differences, which means, put in simple words, he tried to come up with, try to understand when is a grayscale difference visible or not for a human observer. So when you have two levels of grayscale or two luminances of grayscale, when will it be visible, when will it not be visible? And that is actually shown or summarized in the graph on the top left, which shows on the bottom the just noticeable differences, and on the vertical axis the luminance range. So without giving a lecture of an hour on this topic, just a few points that I want to highlight here. It's a non-linear curve. So it means that it's not that simple. You cannot make a statement like, if there is a 3% difference in grayscale level, it will be perceivable. No, it also depends on the absolute luminance level. And the second point that I want to highlight is that the human eye is more sensitive at higher luminance levels. And that has also been illustrated in the bottom here. It's the same pathology image, but one is very dark, but still having the same contrast ratio. The right one is much brighter with the same contrast ratio. As you can clearly see, you can see much more information on the right than on the left. So that's important to understand. Of course, as I said, pathology images are not grayscale images, they are color images. So there has been a lot of research also on perception of color. On the right, you can see the macadam ellipses. So it's a little bit similar as JNDs. What these mean is that within the ellipses, it's impossible for a human observer to see differences between subtle color differences. So the color points in those ellipses are indistinguishable by a human observer. So why does that matter? Well, I'll explain it with a picture on the left. It's 2 times the same preclinical image of a test animal, but with 2 different display settings. So on the left picture, you can see it's very hard to see subtle color differences. If you look to the color scale that is shown, you see that there is very little yellow in there, and that the blue is hardly distinguishable. On the right, it's much better. So this purely has to do with the way that the display is visualizing the colors. So the message I want to give with this slide is the display has a big influence on whether things are perceivable or not by a human observer. So that was a little bit of theory. I hope you're still with me. The next part that I want to talk about is on medical and non-medical display systems. Because they're both displays, they look very similar, but they have completely different functionality, completely different behavior. A few points that I want to highlight here. So if you look to medical display systems compared to professional consumer displays, there are a few specificities or a few special functionalities that you will find in medical displays that are not present in professional displays. The first one being stabilization and consistent behavior over time. So most medical display systems are full of sensors. So they are full of luminance, color sensors, backlight sensors, you name it, full of sensors that guarantee that the display behavior remains stable during the entire lifetime of the display, which can be 3 years or 5 years, typically for a medical display system. Just one example on the left image, the one with the crop with the display. If you look carefully there, you will see a small patch on top of the front surface of the display, but that's an example of a front sensor. So there is a full sensor in there that can actually measure the light, the grayscale, but also the color characteristics of that display as it's being perceived by the user of the display. And it's not only measuring, but as I said, it's also stabilizing. So it really makes sure that over the entire lifetime, it's perfectly stable and it always looks the same. If the display is brand new or 1 year old or 3 years or 5 years old, it will always look the same. The second one is calibration. So I explained a few minutes ago about the importance of correctly visualizing images based on understanding of the human visual system. Well, that's exactly what calibration will do. Calibration will make sure that the images are visualized in such a way that they can be optimally perceived by a human observer and that all the details can be optimally seen. If you don't do that, then it's a little bit of coincidence or luck, whether you can see things or not, and it may differ and it will differ from one display to another or even from different units of the same display type. So calibration is also a very important functionality that medical displays have. Then we have the third one, uniformity and image quality corrections. So medical displays really are full of image processing algorithms. So they do not just show the pixels as they are in the image, but they process the images. They do all kinds of corrections, and my colleague Brian will explain more in the second part of the webinar. They do all kinds of corrections to make sure that the image quality is improved and is optimal. Quality checks and controlled manufacturing. You may say, well, quality, everybody ships good quality. Well, of course, for medical displays, there is a lot of certifications that need to be fulfilled and additional validation and testing that needs to be performed on every individual unit. But also the controlled manufacturing is important because in medical devices in general, you cannot change things to your medical device without revalidating and ensuring that there is no change in behavior. For professional displays, this is typically not the case. For professional displays, they take the freedom to, for example, swap panels and use other types of LCD panels depending on the current supply or price of the panels. So you may actually buy a unit today and you may buy one 2 weeks from now, and they may have different behavior. And then the last one, medical displays really are designed for a long lifetime. So it's not something you need to buy every year, every 2 years. Typically, it's 5 years of warranty. Just a deep dive on the first point, on the stabilization and the consistent behavior over time. If the slides wants to move. Yes, here it is. This is an example of what can happen and what will happen if you do not stabilize your display system. So this is on the left side, a pathology image as it would be visualized on a brand new professional display system. And on the right side is what it would look like, I believe, after 10,000 hours of life of that display system. So a few things you can observe here. The right one is much darker, so you lose luminance, which is important. Remember the statement I made on visibility of subtle details is much higher when there is a higher brightness of the image you're looking at. But it's not only luminance, also the colors are shifting quite a lot. And you can look at the reference that is added here, or you can look at the graphs. There is also quite an important shift of color, of saturation and hue in those images when the display is aging, which is of course not desired, because you would like to see the same image on the display if you look to a certain digital slide today or tomorrow or in 2 weeks or a year or 2 years from now. So that's only possible if you stabilize the display system. I think that was my part of the webinar, and I gladly would like to hand over to Brian.
Hey, Tom. Can I just get a thumbs up that you can hear me?
Yes, we can.
Perfect. All right. Thank you very much. I'm going to take over from Tom and try to take some of the information, the scientific information that he applied, and apply that to what a display that's fit for use in digital pathology is. I think at the highest level to think about that is the display is really the human interface device to the digital image, and therefore it's a very important component of digital pathology. When you go to make a decision related to display, what's going to happen is you're going to get hit with a lot of specifications. The thing is that specifications sometimes are okay in isolation, but often they're used as in harmony in how they apply. And so one of the things that first happens is, what is the screen size, where the diagonal measurement across the screen surface, and then resolution gets mentioned. Resolution is the number of pixels, but those two things appear to be independent. They're actually dependent if you try to look at image sharpness, clarity, or visual acuity. And there is no specification that is for image sharpness. It becomes something that the visual perception that you see as the user. And the eye determines in a lot of ways what resolution is, not the actual number of the individual display. And I'll demonstrate that in sort of the next area. One of the things when we moved into this field that we think is really important to maintain is when you shift from analog to digital, what is the workflow? The first assessment of any slide before it goes into the microscope is just like this. This is the overview of the perception and the individual perception. The number of pixels that are available on the screen and the distribution of those pixels will determine how well this first step in digital pathology diagnosis is really launched. And that begins with really a discussion about pixels. What you'll see as a specification is pixel pitch. Pixel pitch is just the difference between the measurement and the distance between two individual pixels, which you'll find in specification sheets. But when it comes down to a pixel also has to be distributed across screens of different sizes. The number of pixels and the size of the screen determines pixel density. That is not a terminology that's in a specification sheet, but it's something that your eye will recognize, because a pixel in digital pathology is a piece of tissue. The more pixels, the more tissue. The denser those pixels, the more clear and crisp and bright that tissue is going to appear. I'm going to talk first off about this representation of pixel. And this is a slide that's going to build. This is a large resection slide, and it's at 1x zoom factor, which is the native acquisition likely of the scanner that took this. And this is kind of like where digital pathology started 6, 8, 9 years ago. This was a high caliber display. It was a 2 megapixel display at 24 inches. 2 megapixels, I mean, those numbers mean just across the bottom on the horizontal axis, there's 1,920 rows of pixels across the bottom. And in the vertical axis, there's 1,200. That's what you see of this resection slide, but there's more. What happened in the first display that was used, I don't know how fast this builds. I hope it goes pretty fast. But if you go up in size to 27 inch and you get 4 megapixels, you double the number of pixels that is there, and you get to see more tissue because there's more pixels. But if you keep that same 27 inch and you double the pixel amount one more time, to 8 megapixels, I was afraid that's going to happen. I'm very sorry about that. To 8 megapixels, this is the amount of tissue that's available at the same zoom factor. So the theory is, as Tom said, in this discipline, there's a tremendous amount of panning that has to happen. If you can see more tissue on the individual screen, the theory is that you will be able to move faster through that image because there's more tissue that's available. So when you take a look at, go to a consumer store and you look at the screens and the displays and the TVs, you see something a lot like this. There's different colors, profiles, there's different hues, there's different brightnesses, there's different sizes. And the question becomes, well, which ones are those is correct? And in that environment, the one that's correct is really preference. It's preference of what feels better to you, and maybe that time of day or that day of the week, it feels better to you. But is preference really the right decision for a digital pathology display as a diagnostic tool, as that human interface device or human interface with that digital image? Especially as digital pathology moves into enterprise imaging, that will become into question with regards to what is the correct image and what is the consistent image that needs to be seen across the enterprise. So Leeds University in the UK has done a lot of research. They have a lot of webinars, training webinars, and I would encourage you to go out and look at them. But when they made the conversion to digital pathology, this is 2017, they did an actual study. They brought in all different types of displays. They brought in 8 displays that had been positioned as displays for digital pathology. They brought in 34 pathologists. They showed them this one slide, which is a benign nevus slide. And they had those displays, they were all calibrated the same way. They brought in IT experts. They brought in calibration software and paraphernalia and tools. And they made sure that the displays were in the same environment. The image management system had the exact same factors on it. And they even took these pictures at the exact same distance from the screen. And you can see what happened. None of them looked the same. And what they said is, despite the attempts to standardize the appearance of the whole slide image in terms of color, the ability to do so was limited by the technical specifications of each display. So they've accepted that's, especially when you mix medical grade and non-medical grade displays, you see that. But what they also did is they had a visual analog scale of scoring, of preference, not just the one of the display they preferred the most, but they also put the range of what they liked, and they gave some score factors. And from that, they correlated specifications in isolation and specifications in harmony that tended to work. I would point out that the star shows the display that did win. The display that did win has more pixels, has more tissue that's available to be seen. So they preferred that. But compared to this group of displays, which runs a very wide gamut, what was shown as qualities that were preferred is image brightness, image uniformity, things Tom has referred to, image or resolution, which is really just numbers of pixels, which was I've been referring to. And then we get into the idea of, well, when you're replacing a microscope, what is familiar from a color profile, from a crispness of that image? And you take in that in consideration, the medical grade displays really did rank out 1, 2, 3, really all towards the top. So this is display to display Leeds University. Barco is a market share leader in radiology, and we wanted to use our radiology technology and know-how to move into the digital pathology image. That comparison is a little bit different. On the left is a Barco Corona 6MP, which is a 31-inch display. You can see the pixel pitch and the refresh rate. And on the right is the image of a Barco display that we built for digital pathology and brought out a few years ago. It's 27 inches and it's 8 megapixels. But when you compare these two individual displays, there are differences. One, we realize when you move from a radiology environment to a digital pathology environment, you need more color capacity. You need greater pixel density, which is really a surrogate, the right surrogate for resolution. Chromaticity, I'll be honest with you, is a very difficult word. It's difficult to do in front of Tom as well. But chromaticity is a combination of what Tom was talking about, and that it's the luminance or the brightness combined with the hue of the color and what the human eye perceives that. How consistent is it? How correct is it? Refresh rate is also something that the displays had to move much faster because there's so many pixels and so many slide areas that are moving around. And there's a slide on that. Also, relative to a radiology display, screen size needs to be a little bit smaller, contrast ratio has to be a little bit lower, and calibrated luminance needs to be a little bit lower. Still are all high relative to a consumer grade display. But radiology is pursuing a direction in luminance and contrast ratio that is just not conducive for digital pathology. And I hope this loads, it does. Okay, so in radiology, you're looking at black, which is air, and white, this is bone, and you want to have as much contrast as possible to see that individual grayscale. That is not the case in digital pathology. This is stained tissue. This is not a mathematical reproduction. And most frequent stain, 80, 90% of stains, or an H and E stain, which is a hematoxylin and eosin. Hematoxylin stains the nuclei in this case, and looking for mitosis. And if you have to look at mitosis of that cellular division, you've got to determine, is it dysplasia or is it cancer? And that is dark blue and dark purple. And you have to make this discernment for these cellular structures that are stacked next to each other and stacked on top of each other. So when a pathologist looks at this image, what they tell me is it's good, it's bright, but the radiology image is too aggressive. It's not familiar to what they're looking at in the individual microscope. And this is what we strive to achieve, to make something with high performance, but high performance not for radiology, but for digital pathology. This is the next comparison. This, on the left is the Barco display. It's an 8 megapixel display. It's 120 hertz, is twice as fast as any of our other displays, and it's 27 inches. On the right is a non-Barco display, but it's a display built for radiology that has 8 megapixels. And the first thing I want you to really recognize here is, guess what? There's the same amount of tissue that's present. And the reason why that is the exact same amount of pixels on the screen. But the images still look different, and it's not just the contrast that's associated with this. It is the idea that the pixel pitch is bigger with the 8MP for the radiology display. So the pixels are spread farther apart, meaning the tissue that's represented is pulled farther apart. What happens with that is it's bigger. Yes, indeed, it is bigger than the Barco display. Size of the structure isn't that much of an issue, because within the image management systems, you have all of the zoom factors you want at any indication. So if it needs to be bigger, you just increase the zoom factor. But what happens, what I hear when I show this image to pathologists, they first will say, well, it's softer. But then they say, well, wait a minute, it's actually crisper. And so that's what it is. It's more familiar to them because the density of the pixels are closer together, presenting a better visual acuity for a representation of a anatomical pathology slide. I also want to make sure I take a beat here and take a moment, because this is in a conference room. This is a comparison between the two displays at a hospital in the UK, actually. But this is not a typical work environment. The radiology display on the right will not sit in a physician's office. It will sit in a reading room, and it will not just be one, there'll be 2 or 4 or 6. And then they also will have administrative displays that sit to the side of that as well. It is designed to be in a reading bunker and a reading environment. In digital pathology, that is not the case. This display has to have a footprint that fits into the pathologist's office. They don't get new offices when you go to digital pathology. So things have to be pushed aside. Often the microscope stays in place as well. And they only work off of one diagnostic display. So the idea that it's smaller is pragmatic for that. The other reason is that the human eye, the bigger the display is, just like your TV at home, the farther you need to move farther away from that screen to take in all of that visual representation and to understand it. Pathologists are not used to that. Pathologists, I say, grow into the image. They go into their microscope. They get close to what they're persecuting and understanding. So you can get closer on a 27-inch versus a 24, which is a very practical reason and purpose why we went with that size of display. The other thing that we do well that you see and why people say, wow, that image is smoother. And this is something that Tom talked about before. It's uniformity and uniformity correction. And uniformity is just how well the luminance of that display is displayed across the surface of that display. And the easiest way for me to talk about this is to really start in this center here. A consumer grade display that doesn't have much imaging processing, like a medical grade display that Tom talked about, looks a little bit like a flashlight, and that the luminance is good, well controlled, green is 100% in the middle, but its luminance and its reproduction of that luminance falls off by 20, 30, 40, even more than that of a percent as you get to the edges of that. There is a manufacturing technique that's used in higher level consumer grade displays that actually tries to dampen out and provide that uniformity. Isn't full uniformity, it gets a little bit lower, but it does make it uniform. But that technology tends to have this problem as well. And these are bleeds at the edges. Those do affect your eyes, and they're not ideal in the situation. So you move into a medical grade display. Barco, as a medical grade manufacturer, we have a lower end and a higher end. Our pathology display is a higher end display, but it has to do with pixel management. And this is a display algorithm, is that think of pixels of a display that are broken up into individual zones on the horizontal axis and the vertical axis. And those individual pixel zones communicate with each other and explain to each other what's going on, so they modulate out and manage that uniformity. You can see that it's there, much smoother, much better than this sort of hilly environment, but it still falls off at the edges. What we did with our high-end radiology displays and what we do with our digital pathology displays, we manage the luminance and uniformity pixel by pixel by pixel. So each pixel is talking to each other to maintain a controlled and calibrated luminance. And you can see even with that, the challenge is always at the edge, and luminance affects your perception of color. So we fall off a few percentages, but it's really an excellent quality. And so that's another specification you can't put on a piece of paper, but you see it when you experience the display that's of higher quality versus a lower quality display. In the interest of time, this is really the same image. It's just talking about in a more commercial way. Per pixel uniformity allows the noise that creates in a typical image, and it manages out that luminance so the colors are more vibrant and more fine details. But as Tom said, I think what he said, and I really want to go back to this, says it makes more details visible and patterns easier to see. That, by definition, is what a pathologist does. They look at morphological structures and they look for patterns, and often they have to count things as well that are associated with it. So that's a key sort of component of a visual acuity that comes with a higher caliber medical grade display. Okay, back also something else Tom said. It has to do with color profiles. As you're looking at displays, you're going to be hit with color gamut. And the most popular color gamut, if you look at consumer electronics currently, is a DCI-P3. And that is a color standard that the movie industry created, because it's deeper into the greens. And if you remember what Tom said about the macadam elliptical effect, to see differences in greens of forests and meadows and fields, you got to put more green into the picture to discern the individual differences. It doesn't have a lot of application in medical nor in digital pathology. You will find it as a specification in our sheet only because we had to have a measurement so that you could put it there, but it's not relevant. The same with the Adobe sRGB. Also went deeper into the greens, but also deeper into the blues as well. They needed that for the advertisement industry. They needed that for the print media industry as well. But for digital pathology, what you see is the sRGB gamut is what the whole slide imaging scanners are looking for, what it reproduces. Take a very close look at that number. It's easy to get to 96, 97, 98 coverage of sRGB, but for a liquid crystal display, it's very difficult to get to 100%. That's separation between low quality and high quality. But as Tom said, outside of that gamut, in this expanded hematoxylin and eosin, these deeper pinks, purples, and magentas, there is information there that could be valuable. And with our display that we brought in for digital pathology, we tweaked the gamut into getting into that area. Now it has two settings. I really want to be clear that sRGB is a very standard setting. It's what is expected for the display and is common for the display. But we were able to tweak the gamut and try to reach down into these areas where there's other information on the H&E slides. And this is a two representation that builds pretty fast. And one is so exaggerated that especially when it's my display and your display in the webinar, this is a very good image. This is what's expected. Pathologists are very impressed with that image. This image here is a wide gamut. It is expansive. It is a little bit overly expressive. It always accentuates. But what does it accentuate? It accentuates the eosin stain, which is the stroma or the connective tissue. So it's just not nuclei or lymphocytes to be looked at with the purple. The stroma has disease in it as well, and this can help some pathologists or aid them in seeing the disease within that area of the individual stain. Tom talked a little bit about this, the pyramid. You mentioned the pyramid. Basically, it's one big photograph that the digitized information gets put up layer by layer by layer, and you have to navigate across and through that. Because of that, slides and data is brought across the stream very, very fast. So what came into digital pathology is something that's same coming into the gaming industry as well, is they make them twice as fast. The hertz or the refresh rate of 50 to 60 is common. What this means is that image is refreshed 50 to 60 times per second. It's doubled with a gaming display, doubled with our display as well. But I want to pause right there real quick, is because it's not just how well the surface of the area goes, it's the pixel changes, because these pixels frame to frame have to also change color as well. And so there is technology involved in displays that are medical grade that say, I'm going to change from one color to the next while I'm doing this frame change. There's undershooting and overshooting that happens. Without technology in there to manage that pixel transition by color, it doesn't look so well, or it has an effect on this, more effect on the blurring. But more importantly, our product has a technology, we branded it Rapid Frame. But I want to be very clear, medical grade displays of high quality have other similar technology as well. The point is, it's not just a gaming display or something that's fast. Look at the technology that's associated with pixel transition for color. Medical grade displays have that imaging algorithms built into them. All right, as a medical grade display, Tom hits on some of these points, but I tried to get it all on one page. It's like, what is really the big difference? One of the big differences Tom talks about is that a medical grade display has an embedded optical precision photometer. That photometer is made to calibrate that display. That photometer is made to monitor that display while it's in use so that you know that you have correct quality assurance. It also can be used for some level of remote diagnostic, but it's just not done in isolation. It's done in combination with an enterprise level QA software that manages the policies across that display and its settings. Unlike a consumer display, this is a precision medical device that's used in an ecosystem that's designed for diagnosis. And so it has to have this other software associated with it for it to be at the correct caliber. Therefore, it is not an extra charge. It is a free thing that Barco provides. What I also tell people is that the display, a smart display, doesn't just look at itself, it looks at the environment in which it's operating. And that's an incredibly important differentiator of this display, is that above this, and I realize you can't see my cursor, but above what the little bump out there for the eye guard, you can see another little open sensor. That sensor is an environmental light sensor. Calibration for color is determined by the setting, the ambient light setting of the room. If too much light comes across that screen, it is known that critical structures can be met. And I'm going to show you a little more about that on the next slide. Per pixel uniformity is something we've already covered. And then as a medical grade display, yes, they do cost more. Yes, going through that regulation standard and having a controlled regulated device does cost money, but it also builds safety, efficacy, and consistency into that product. So our products comply with international medical device regulation standards for developing, and we do usability testing with actual users before we release a product. We do cybersecurity testing, we do image management, have a quality management system, and we take all complaints and we track complaints and we manage complaints for the lifetime of the display. And that's not just 2 years or a year and a half, that's actually up to 10 years or even more that we will track a display. We have a quality management system that is mandated and directed by the FDA and ISO, and our factories are subject to inspection and approval and findings by the FDA and by also notified bodies in Europe. Not good. So about the QA software, yes, it maintains the device, but there's some really interesting things that it happens that it maintains. And this ambient light warning is one of them. This ambient light warning, this is a picture of what comes up on the display if in fact there's too much light that's coming across the display. And as I mentioned Leeds before in their webinar, Dr. David Brattle is his name. He has a saying, and it's webinar number 7. It's one of their last ones. He says, you can spend hundreds of thousands or tens of thousands of dollars to get the best display on the planet. But if you're not sensitive to the environment in which it's used, you've really wasted your money. He says it a lot more colorfully than that. But the idea that there is too much glare and too much light on the screen is something that does definitely need to be paid attention to. It's what a medical device does, or at least ours does. The other thing that Tom mentioned is International Color Consortium. It's a topic all onto itself, but I guess think of it this way. Any device that reproduces color through the International Color Consortium and an ICC profile generation can characterize that says, hey, I'm a device. This is how I see color. That can be quantified and provided over to the operating system. So whole slide imaging system can do the exact same thing. It says, hey, I see this color. This is how I represent this color. They create an ICC profile. Both of those ICC profiles are handed over to the image management system that has a color management handshake between the two so that you know what's being displayed on the screen is in harmony throughout the whole systems that are managed in that pixel pipeline related to color. Image quality and consistency are going in, but with regards to this ICC profile, I want to give this quote because one of the drivers of digital pathology is clearly AI. It has application for pathology, and it says, looking to the future, applying ICC color profiles to whole slide imaging technology will also aid in the application and validation of artificial intelligence algorithms. Really what that means is you look at an algorithm, you look at a Gleason score algorithm, you say, oh, these different colors are supposed to represent different Gleason scores, but they are very defined of what they're trying to say. Look here, don't look here, score there. So any refinement you can do to that color will help the AI algorithm representation. This is what we mean by consistent image quality. As digital pathology moves into enterprise imaging, there will be policies associated by the administrators that oversee all of these devices, that this cloud-based AI system or this QA system is our QAWeb enterprise representation. But what this image is really trying to say is regardless if you're a large hospital, medium-sized hospital, or you're connecting remotely, which is a big driver and a big change within CLIA and the laboratory through secure remote connections, you can be assured that all of the calibration of these devices are in calibration per policy, and if they're not, there's a warning sent to the administrator that something needs to be addressed. That is important. What I also personally think is important, as you point out, as a consumer as well, is that this physician, when they are making a diagnosis, there is a record that that device is in calibration of what's expected for them to make that diagnosis. There is a record that is associated, and it's held in these QA systems that are on board. Oh, all right. The lag is right. So one of the things as you get into digital pathology, what's going to find out is that the mouse is not really adequate for most physicians. I know many pathologists that use actually 2 input devices at the same time, looking at input devices because there's so much image space that has to be scanned, and there's so many ergonomical worries. There's something that's going to be part of everybody's transition over to digital. Very much so. So what we did to show sensitivity to that in application, either used as full-time or as adjunct, we put a small touchpad associated with that. It is our touchpad. We designed it for digital pathology. We designed it really for this picture over to the left. Pathologist moves a very small millimeters to navigate a slide. You go on a large display, especially large display, there's a lot of grabbing and pulling. It's designed to use the finger in the thumbnail area of the slide to navigate all of that slide, but only small motions. Other areas, there are 6 programmable buttons at the top that can program certainly like zoom factors. Certain subspecialties only need to look at like 2 zoom factors for all the slides, so they can be pre-programmed at the top. You can all pre-program things like next slide. And then common programmable things that are in there are things that you would get on your smartphone or your other tablet of how to pinch and zoom and change magnification or the zoom level factors. Okay, so I'm going to go to the conclusions. I recognize we're 8 minutes out before this should end. How do you conclude this? What we've concluded is we looked at all of these things. This is not a commercial. This is an educational program. But we looked at all of these elements of what is needed in pathology, and we brought something to the market a couple of years ago. It's very bright. It has a color profile that's incredibly impressive. We have workflow tools on the right, like Focal Path, that we haven't talked to. But as a medical grade display, it stays consistent. It has a 5-year, even an extended 7-year warranty that's available. It has a very impressive color profile. But more importantly, or most importantly, we submitted to the FDA. It is the only display that has FDA clearance for primary diagnosis with multiple whole slide imaging systems, meaning it is interoperable. And the details on that are really this. Of all the whole slide imaging systems that have primary diagnosis comes from the FDA, they all have the exact same indication of use for digitized histopathology slides. This display has that exact indication of use, and there's a validation process recognized by the FDA to say that it is validated for primary diagnosis. So the Philips systems with their viewer and the Page viewer, the one Leica scanner that does have FDA clearance for their viewer, or the Sectra Enterprise viewer, it's compatible. The latest one is the Hamamatsu NanoZoom S360 ND. It has compatibility associated with this. All of those individual systems have an individual display, that individual and only that display. Many of them are now end of life as well. they have a display that's associated. If this is the only display on the market that has FDA clearance to be compatible with all of those, we also have a system in place to maintain compatible as new scanner manders come on board with an indication of use for primary diagnosis. And there are 2 that we know are that are coming actually quite soon, and they will have indication with this NDPC 27. Lastly, when you, as a company, when you make a need, make a product, you have to say, what is the challenge? What is the challenge that you're trying to address? Digital pathology, or pathology in general, and oncology in general, has a productivity issue. The workload is continuing to increase from just the number of scans in the population that has cancer growth. And so the Australian Department of Health looked at anatomical pathology as a workload issue. And they said the percentage of pathologists that are experiencing adverse events, and adverse events is not a misdiagnosis, it's just something that's not done correctly or something that's missed. It could be misdiagnosis or just something that isn't consistent. Those adverse events increases significantly when the workload hits 39 hours a week. And you can see by the graph here, whether it's this is no adverse effects happen. This purple is 1, this is 2, this is 3. If you look at, or 4, if you look at what tends to happen, the difference between almost a 40-hour week and a 60-hour week, there's a tripling of the number of adverse events that's happening. So that was really our objective. It's just like, how do you help the physician with its individual workload so they're not overworked and you don't have these adverse events. Go back to the conclusion for the goals and objectives, really is to understand the relevant properties for digital pathology images, to understand the influences of these image properties on perceived quality from the user interaction. And then my job was a little bit to make the link there to display specifications. And I tried to be really clear is that it's not just 1, 2, or 3. It's most of them working in some type of an amalgam of a concoction of things that make a good image quality, makes it fit for use. And for us, fit for use is something that actually improves the speed to diagnosis, improves the confidence in the diagnosis, and improves the reproducibility in diagnosis. And that's what we set off to achieve when we made a purpose-built device, or this human interface device, specifically for digital pathology. Through Barco and through SIIM, this is an educational program. As an educational program, we want to make sure within the record, we want to get the references that are there that we use today. That was 1, that was 2, really quick. I want to make sure that they are recorded. And that's 3, unless Andrew's driving these slides and not me. But anyhow, I want to make sure that those are there. And then the next slide is I'm going to give back to you, Andrew. I've been talking, I haven't looked at any of the questions that have come in.
Yeah, so if you guys look in the Q&A section, Tom has answered a few of the questions already. So Tom, you want to maybe kind of quickly browse over kind of the answers you just replied with?
Yes, of course, Andrew. So there were very interesting questions. Thank you for that. So there were questions, for example, on the pyramid structure of the pathology images, whether all those different resolution levels actually are present at the same time in the file. So the answer is yes. Mostly the scanner formats include all those different levels in one and the same file, so that you can quickly zoom. There was an interesting question on off-axis viewing. So if you have multiple displays next to each other and you look under an axis to one of or multiple of those displays, whether that's a problem. So the answer there was no. With high-end modern medical display systems, we pick very high-quality panels that allow for slight off-axis viewing, and in typical viewing conditions, this will absolutely not be a problem. Then a very important question on whether there is going to be a need for pathologists to go sit in a special reading room. So I explained in the Q&A that in the past this was the case for radiologists, mostly because long time ago, the first medical displays were quite dim. And because they were so dim, the only way to be able to perceive all the image details was to go sit in a darkened environment. With modern medical display systems, they are high brightness, and you can actually read in normal or almost normal office conditions. Yes, I think these were the most of the questions asked so far, but there are a few open ones. Maybe Brian, I think there is one on ambient lights and impact on the monitor and how the monitor takes that into account. Maybe you can answer that one if that's okay.
It takes it into account. I covered it in the presentation. Maybe the question came in prior to me covering it. I think the important part is to say is the calibration of a display, a color calibration display, starts with understanding what ambient light was in that room. And if it exceeds that ambient light, the calibration is not considered correct. I would encourage you really to go look at that webinar because they show in the webinar a representation of how too much ambient light actually affects being able to identify structures on the screen.
And then there was also a question, Brian, on whether the touchpad comes with the monitor or whether it's separate.
Yes, it comes with the monitor. Apologize, I was looking at the clock the whole time. It's like you're going to specify. Yes, it comes with the monitor. It is standard. We recognize that it is an important element to have some sensitivity. I've given it to pathologists, and I've learned more about it from the pathologists. And it comes down to really very much so trying to make very small motions, and it enables that. It may not be a full-time device, may not be for everybody, but it's worth really a try, and it's very useful. It's designed to fit into a pathologist's office as well.
Okay, I'll just kind of wrap up here since we are almost at time. So as you discussed earlier, attendees must attend for 45 minutes to earn the CIP ABII credit. If you have any questions, please email the SIIM group at info@siim.org. There's also some new SIIM training courses. Here's some upcoming virtual offerings. And here's also a call for session proposals due by August 10th, National Harbor, Maryland. And please take the post-webinar survey. Help SIIM plan for future webinars. And definitely thank you very much to the group, and thank you to everyone. Thank you to Tom and Brian for your time in presenting this information. It was really, really good content.
Bye. Thanks.
Take care, everyone. Thank you.
Thank you. Bye.